Have you ever wondered how an AI model can take a look at a 3D microscopy video and figure out where the cells are? The answer in some pipelines may lie with the temporal unit. Huh? But how does that work? Let us start with the raw data. A microscope gives us a time lapse. That is a sequence of 3D images showing cells changing over time. We can think of each frame as a 3D volume, and the fourth dimension is time. So overall, we have three dimensions of depth, height, and width changing across time, right? But before the neural network can digest these images, we need to prepare them, pre process them. The most important step in this pipeline is to normalize the intensities. Some movies may be taken in high intensity, some in low. The model does not need to learn on different intensity sets. So, what we do is that we normalize them. The model gets to learn from one normalized intensity range. The next step is to downsample these images in X and Y. This is just done to reduce the amount of computation. The X and Y are much more in number, the X and Y values are much more in number than the Z values. And so our resolution decreases and our data becomes more digestible to the model. Now the prepared frames go into the 3D temporal unit. But why a unit specifically? Well, we can use other things, but a unit doesn't just look at the local image details. It also builds a broad understanding of what the 3D surrounding structure is like. It uses three dimensional kernels to build a complex feature map, and that is one of the outputs that it produces. The second output is detection logits. The feature map are the model's representation of what it thinks the image or the video is. Very simply put, it is what the model thinks a cell is supposed to be. Uh, it is not just three dimensional in nature, it is multiple things, but this is a representation. On the right is a detection probability. That is the bunch of pixels the model thinks are the cell. But we cannot have a cell occupy multiple pixels, right? If we are to look at a cell, a singular item, we have to give it one XYZ coordinate. So 1 pixel. For this, we identify the local maxima, the place where the highest intensity score is or the highest detection score is, and then we classify that as the coordinate of the cell. And so we've done it. We took raw microscopy data, we gave it to a neural network, it gave us possible cell candidates, and we represented where the cell is. That is the first part of cell tracking.